Good morning. This is episode 2, why reverse phase chromatography RPC is used in pharmaceutical analysis. RPC is used in 70 to 80% of all HPLC applications. And here are 5 reasons why RPC predominates in the analysis of small molecule drugs. Reason number 1, reverse phase has great retention for most small molecule drugs. Reverse phase retained most small molecule drugs from slight to moderate hydrophobicity. In the full gradient, most small molecule drugs elute at about 60 to 70% mobile phase B, such as acetylnitrile, because very hydrophobic molecule or very hydrolytic ionic molecule are less likely to be good candidate for orally available drug products in small molecule arena. Next reason, reason number 2, reverse phase has 100% mass balance. The retention mechanism in reverse phase is primarily something called hydrophobic interaction, which happen to be a weak interaction. Therefore, the column in reverse phase is less likely to be contaminated, very much in the case for normal phase chromatography, which is totally ineffective for analysis because of column contamination. Also, all components in the sample under reverse phase, because of weak interaction, are eluted from the column under strong mobile phase condition at the end of a full gradient. This situation for 100% mass balance is a critical requirement for stability indicating or purity assay, where all the components of the sample is supposed to be eluted from the column. In this diagram, we show you retention mechanism of hydrophobic interaction, shown here with a silica particle with ligand, which are very hydrophobic, such as C18 ligands, and with the mobile phase of aqueous mobile phase, where nonpolar particles are retained and also polar particle eluded first. The retention mode is called hydrophobic, or a better term is hydrophobic interaction. The primary retention mechanism of reverse phase is hydrophobic interaction, or co-partitioning of the analyte into the hydrophobic stationary phase with something called a dispersive forces. Hydrophobic interaction is indeed a weak interaction, and therefore it's unlikely that column would retain any component from the sample after elution with strong mobile phase B, leading to near 100% mass balance. Perhaps the term solvophobic interaction is a better term, since there are some interactions of the mobile phase component, which consists of buffer, water, and organic solvent, and they play a role in both retention and the selectivity of the analyte under reverse phase condition. Reason number 3, reverse phase is very compatible to gradient analysis. In gradient elution, typically use much higher peak capacity and also offer better resolution of components of very diverse hydrophobicity. Also, column equilibration is very quick for reverse phase, unlike in the case of helix, which is 3 to 5 times slower column equilibration, allowing reverse phase to be highly effective in high throughput screening. Finally, reverse phase offer excellent long-term assay and column lot-to-lot reproducibility using column packed with bonded phases from high purity silica support. A note, most critical pharmaceutical assay use gradient analysis. In this slide, we demonstrate reverse phase gradient profile and some terminology. Shown here is a gradient profile, which is a very generic full gradient from 5% to about 100%. The gradient is linear, completed in 10 minutes, and that's called gradient time, or T sub G, which is 10 minutes. The gradient range is 5 to 100% mobile phase B. As indicated, most small molecule drug will elute from 10% to 60 to 70% mobile phase B due to the drug-like property of slightly to moderately hydrophobic molecule. If a drug is too hydrophobic, it would not be a good orally available drug candidate. Typically, we have a first step at very high mobile phase B, and then a ramp back and equilibration. The entire assay time shown here of the complete cycle is 16 minutes. So once again, this is a generic gradient method for sample screening. For instance, 5 to 10% B in about 10 minutes. In the ballistic gradient for high throughput screening, typically we do the same gradient profile completed in 1 to 2 minutes. Total analysis time in the cycle is 16 minutes. I mentioned that the reverse phase RPC gradient analysis we offer very fast equilibration and also very flexible method fine-tuning for method development. For further information, check out episode number 4 on reverse phase mobile phase fundamentals, shown here in this YouTube video link. Reason number 4, RPC allow very effective selectivity fine-tuning for the development of stability indicating assays. It offers many factors for effective HPLC method optimization. Mobile phase factors such as pH, buffer, organic solvent, and the use of other additives such as ion pairing and also chaotrops. There are also many operating and gradient conditions that can be optimized, such as flow rate, temperature, gradient time, which is pivotal, gradient range, either in a single gradient or multiple gradient segments. Finally, many very effective column selection choice, starting with C18 to C8, which is very similar, cyanol, phenyl, pentafluoro phenyl group, and also polar embedded bonded phases. Further information can be found in episode 11. Reason number 5, RPC is very mass spec compatible. RPC using volatile mobile phase additive is highly mass spec compatible with both ESI and also APCI, electrospray ionization, and atmospheric pressure chemical ionization. Mass spec compatible methods are really important for peak identification and quite desirable for early phase HPLC method during new drug development. Further information is found in episode number 17. This slide captures some major shortcomings of RPC, which is a lack of retention for highly water-soluble compound or highly soluble drug. And there are several mitigation strategies shown here. First is the use of specialized RPC column for more water-soluble compound. Some example are Waters Atlantis column, polar embedded phases, and any column labeled AQ, which are typically equipped with polar end caps. Next is the use of high pH mobile phases, particularly effective for the analysis of opaloids, which is very water-soluble basis, because at high pH, about 8 to 10, these are non-ionized, are neutral for good reverse phase retention. The third strategy is ion pairing chromatography, found lately in ion pairing using TEA, triethylamine, or heptafluoroisopropanol for the enhanced retention and separation of oligonucleotide drugs. Finally, one can switch to HELIC or hydrophilic interaction liquid chromatography by using a silica of diol columns, very effective for the analysis of glycans, sugars, amino acid, and also secondary metabolites. Here are some references. First one shown here by Snyder, Kirkland, and John Dolan. Second one shown here is HPLC and UHPLC for practicing scientists, published in Wiley in 2019. And also YouTube channel shown here for HPLC for practicing scientists, a series of 24 videos. Next video episode, why do we use acidic mobile phase A? How do we reduce peak tailing for basic drugs? And why don't we always need buffers in reverse phase chromatography? On this note, we thank you for your attendance for episode number 2, why is RPC used in pharmaceutical analysis? And we thank our channel collaborator, LCGC, and please subscribe for later videos on different topics. Thank you for your attendance.